Joanna Jackow, and I'm group leader based at St. John's Institute of Dermatology, King's College London. And I have started my own group more than 3 years ago with the spirit and ideas of trying to pursue some new avenues to treat rare skin diseases with precision therapeutic gene correction using DNA genome editing. And today I have a pleasure to present some of our progress and ideas that we have moving forward for therapeutic application in this direction. So just to briefly give some flavor of the problem that you're most familiar with, I'm showing this picture to show the complexity of the skin on the molecular level, including all the proteins that are interconnected and so important to bring our skin together. And as you all know, if this is somehow defected, then the problem starts. And this begins with some disconnection between the epidermis and the dermis and the depicted phenotype of the skin, which is shown here. And I will, on example of dystrophic epidermolysis bullosa, which is most familiar with me, as this is the model I have been working for many years on, is related to a pathogenic variant in collagen 7A1. And as you can see here, in a normal skin, the gene looks quite normal, is structured and fold in propagated way. And only one base pair can be changed, which can be the C to T, and this gene can be completely dysfunction, leading to the blisters of the skin, the disconnection of the epidermis to the dermis, as you can see on this electron microscopy picture comparing to the normal skin. So there has been avenues over the last, I would say, 20 years that scientists have been trying to pursue and have done amazing progress in these technologies. But I will focus on the work that we're doing in the lab, which is the gene editing. And one more time, this is the technology in which we're able to basically somehow fix these mutations to the normal sequence, and then giving the potential of cure some permanent restoration of type 7 collagen. And I will, within my talk, I will try to simplify it and explain you the concept behind this. So just going back to some biology lesson and chemistry lesson, I will not explain anything in details, but just want to make sure that you understand what genome editing is. It's basically some process that has been developed to interrogate with our DNA directly. And maybe you all remember from some lesson in the biology class that the DNA is structure of bases that pair together, and we have adenine and thymine, A and T, and we have C and G that always comes together. And now scientists have used this knowledge and developed technologies in which can interact with this binding in the DNA in the way that we can basically change these bases, basically replace some bases and make deletion repair or insertion repair. So everything what is shown here basically leads to develop these genome editing strategies, which are depicted here in this slide, which, for example, one of the strategies, and this was the first one described more than 11, 12 years ago, allows us to basically cut some pieces of the DNA and then replace with the healthy DNA. We have more elegant methods called base editors that basically allows us to erase some mutation. We call it like point mutation can be exchanged to bases that we want to have. And even more sophisticated but elegant method, which is called prime editing, allows us with a very elegant way to correct with the erasing of the gene, the insertion and deletion and single nucleotide conversion. So we're very excited about this, and that's how my lab has started to work on and provide some knowledge and evidence for a therapeutic potential of this. So, but more recently, and this is something that excites us a lot, is new technology based on genome editing allows basically to replace the whole gene of collagen 7 on any other genes related to skin, as not only to skin, in a way that we basically can program this machinery to install a copy of the genomic or also the copy of the collagen 7 into the place where it is normally located in our genome, giving the possibility to basically naturally re-express this protein. This is some method that can provide very universal treatment for all patients because it's not mutation dependent, but can be done permanently. To make you visualize this a little bit simplified, it's basically like installing the DNA in a specific runway sequence. So we can show that you have one runway in an airport, and then you bring the collagen 7 elegantly in a position where you want to have it, and that will be giving the ability of the cells to produce permanently the collagen 7. And just to conclude from what I have said so far, this all, and especially the paste method, which we know extensively working on, will be very universal for all patients. It's not patients independent from patient's mutation, as also can give a permanent cure because can be installed specifically in the genome position where we want, and ideally it will go in the same position where our endogenous, the collagen 7 that you already have in your cells, is placed. And this is our ongoing research on establishing the molecular basis for it. Compared to, as you all know, to the BVEC strategy, we know that BVEC is also universal and independent from mutation the patients have, can be supplied for the patients, but it's not permanent cure and can be repeatedly applied to the patient's skin. Effectiveness is revealed. And coming to our to the genome editing strategy that rely on single mutation correction on deletion, it is a very elegant way and can provide a permanent cure, but is however not universal, which in some cases can be very applicable because there are a lot of mutations that are common mutation and is smaller, more elegant method to be applied. So, but is just the overview of the spectrum of possibilities we have at the moment. So, therapeutically, the genome editing has some advantages that we have to point out, especially specifically that the reversion of the pathogenic variant is happening on the genomic level. So we not worry about to express too much collagen 7, or there can be some insertion mutagenesis problems related to it. We will have permanent correction with restoration of normal physiological level of the protein, and there's less risk of other new mutation that can happen, and also some adverse effect with immune reaction related to the protein re-expression. And technically, this genome editing strategies provides a variety of options for the therapeutic product, which I will explain you in a moment. Exactly on this slide, you can see how you can deliver and how this basically can be used for some ideas of therapy type. So all these editors or the genome editing tools that I mentioned, which are the scissors or the pencils like base editor and prime editor, or the paste, they can be produced as molecules. And these molecules can be either plasmid or protein or mRNA. And these all have some chemical structure that can be delivered into the cell differently. I will not talk about this, all these possible options that we could imagine today, but I wanted to focus on what we're doing in the lab, particularly, which is the mRNA delivery system. With we're working on base editor, on prime editor, on the paste, which we are using in form of mRNA, in a purpose of to deliver it to the cells via lipid nanoparticle, which are specific structure that are easy to make and formulate and can easily basically encapsulate our mRNA and then can be given to cell types for, in our case, are these fibroblasts, keratinocytes, and skin stem cells in a form of either some injections or topical treatment, basically on the surface of the wound or the skin. And now I will give you some example to convince you that what we have done so far makes sense and really provide us evidence on some therapies going forward. So to start with, we have been using some of the editing tools, and here specifically base editors, which are the genome editing pencils that can basically correct the point mutation in form of mRNA. And you can see here from our patients cohort, we have used patient cells that have heterozygote mutation, and the one mutation of interest was C2T mutation. You can see here this mutation show us like a double peak because it's heterozygous, only one allele is mutated. And we were interested to show that this red basically mutated signal will be completely disappearing. And this is what happened when we add to the cells the base editor called adenine base editor. And we sequenced this and we could confirm that this is real. And also on protein level, we could demonstrate that this protein from these corrected cells can be deposited at the dermal epidermal junction, basically in the skin between epidermis and the dermis. So we have proved some principles that this molecular base of this genome editing is functional, and we can have some benefit for the patients, for the cells. So we then move on further investigation on correcting more mutation, testing now not only heterozygous, but we move to homozygote mutated patient cells and that have a biallelic collagen 7 variant. And you can see here, this is the patient cells with the mutation in green, healthy control in red, in black, excuse me. And when we apply this editor to the cells in order to correct this green back to black, you can see here as more of the editor we're editing, like this is the percentage of the 10, this is the percentage of the editing, depends on the amount of the base editor we have added to the cells. We can show that basically this black peak will be completely present at this amount of the base editor we added to the cells. Very encouraging, and we have sequenced this multiple times, also confirmed with more sophisticated methods such as next generation sequencing that this sequence is correct, and also this mutation correction is permanent, is not going back to the initial sequence after several passages. So this work has been done in the cells as a proof of concept. And so meanwhile, we wrote the grant that is now under support of DEBRA about the paste method, and we're making a progress to hopefully show that also the paste is effective, and we not only can correct the point mutation, but also we can exchange the whole collagen 7 gene. And yeah, this work have just started a couple of weeks ago, and yeah, so hopefully sometimes we will have some updates on that. And meanwhile, once we've ensured that these genome editing technologies bring some potential of correction, we thought what would be the therapeutic strategy that we would like to liaise with these genome editing methods. And we have done an experiment in our laboratory in which we applied mRNA encapsulated in this lipids, in some lipids, and we have basically applied to skin construct, which are some construction from cells in a 3D model, and we can basically reconstruct the normal human skin or also EB skin. And when we did that and apply mRNA that codes for a green protein, as you can see here, we could demonstrate that this is penetrating through this skin and we could see some green cells. And this gives us some ideas that maybe this is a way we would like to go further, and then lies this topical genome editing as a therapeutic using lipid nanoparticle, as we call it, maybe a gene cream for genetic skin diseases. And this is a nice article wrote by my one of my PhD students that is in a really simple way of explaining how skin work and how this therapeutic genome editing via lipid nanoparticle could be used for therapeutic applications. So if you're interested to gain a little bit more knowledge in this, please have a look. So lipid nanoparticles are basically vehicles. So you can imagine this is some like structure like this, and then inside these little vehicles, you can basically have your mRNA, and this is what we're mixing the mRNA with these lipids as two liquid together, and then we can basically then placed on top of our cells or skin constructs. And simplified, this is what we're doing. And one of the experiments that demonstrated the visibility of using these lipid nanoparticles in cell culture to correct the gene is demonstrated here. And you can see here, this is the fibroblast on top of which we have basically encapsulated here to visualize this very well. Again, the green encoded mRNA with these lipid nanoparticles, and then we have given this to the cells. And you can see here that the proof that this works shows us that the GFP starts to be expressed in the cells. So that means this protein have been delivered via mRNA into the cells and then started to re-express. And that was our first experiment, and then we moved to take the lipid now with our editing tool. In this case, it was the base editor that corrected the point mutation that I showed you before. And again here, this is our normal human fibroblast, like a wild type sequence. We have patient sequence with the double peak here, as is shown, as I showed you before. And when we deliver this base editor with the lipid onto the cells, we basically reverse this red peak completely to blue, and is very similar to how you can see here in these corrected cells. So it's very, very encouraging. This has been also several time proof, and yeah, we actually have written some small article about this, and is now in press. I think you could also have a look in more details about what we have done. And lastly, now the trying this proof of concept in some real skin, we are constructing the 3D organotypic cultures, which are composed of fibroblasts and keratinocytes, and they cultured in transwell, some basically boxes, and you can then feed them like with some media, and you incubate them in incubators at 37 Celsius, and you basically grow structures like skin. And we use this as a very good model for what we want to test with this topical formulation of the lipid encapsulated with some of the mRNA. And this is one of our, again, proof of concept studies on maturated 3D skin construct with more thinner and more thicker epidermis. And you can then see that some here of the red signals demonstrate that this mRNA that encodes for red signal has been delivered to the cells and is expressed here even on the basal layer of the epidermis, where we have our epidermal stem cells present. So this is very encouraging, and we are very excited. So we're doing more tests using this model, as well as we're moving on some mouse experiments on a mouse that is also suitable for test this base editing strategies and other genome editing strategies. So hopefully there will be more data in the next few months to share with you. And with this, I would like to acknowledge all my amazing team who are very dedicated for this project that we're running in the lab, and without their help, this could not have happened. and my PhD students and postdoc Karina Graham, who's now basically supported by Debra for the PASTE work, and the founders that supports our work like Debra and QRB, EBRP, Wellcome Trust, and our collaborators from the medical side as well as from the research side within London area as well as outside London and in US, in other European countries. With this, I'm thank you again for your attention, and I'm happy to take any questions if you have some. Thank you.
Thank you, Joanna, for that talk. Are there any questions?
Hi there. Your research sounds really interesting and exciting and promising. I wondered if you have any idea when you might end up getting to human trials?
You mean time-wise?
Yeah.
Yes. So yeah, that's a good question. And so we are now approaching the phase of like some animal work, preclinical, still on preclinical level. So I think another 2 to 3 years will be needed to collect all the required data that we need to provide before proposing some first clinical trial.
Thank you.